and also I will write now a little bit bigger. Yeah. Is this well visible for everyone? What I wrote here already here on the left? Okay. Otherwise, if this is the problem that you can't hear me well or can't see it, please let me know. Yeah, anytime. Good. Because I couldn't finish lecture 1, so today the goal is to continue with lecture 1 and then to jump into lecture 2. But the only thing I would like to, Ricardo, could you maybe add this HDMI cable, because it's not here, to connect to the beamer. I need the HDMI cable connection. Yeah, okay, thank you so much. It's on the other side. Okay, thank you so much. But I would like to continue with the chalkboard talk, and first of all, just give a short summary. So I showed you yesterday, let's see first of all whether that works, how to derive this free energy. In the literature, it has also a famous name. In particular, when n is not 1, then it's called the Flory-Huggins free energy, because it goes back to 2 very famous physicists, and this is Huggins and Flory. And it has these 2 contributions. This is often said to be something like a negative mixing entropy. So as I told you, when you think about the lattice, you just ask how many configurations do you have to distribute particle A on the lattice. You just count the number of these microstates. And then if you do this with Stirling approximation, you get exactly this contribution. And the second one is actually an interaction term. Yeah. And chi is an interaction parameter. So whether it's positive or negative will determine whether the system can phase separate. And when it's positive, actually, when you think about the definition, it comes with repulsive interactions between A and B, and or attractive interactions between AA or BB. And then what we showed is then when you plot this function, phi, f of phi, you get here this beautiful double well potential. And as we know that there's a regime here that is concave, in this domain, the homogeneous thermodynamic state with a constant phi cannot be stable because the isothermal compressibility is negative. And this can't be true. And the way out is to do a Maxwell construction, or in modern times you call this convex hull, that connects here the free energy with a coexistence solution. Mathematically, once you minimize the total free energy, we obtained 2 conditions, yeah? And they describe what is typically called phase equilibrium. And if we have no other processes, for example, no chemical reactions, this phase equilibrium is equal to thermodynamic equilibrium. And it amounts to equal exchange chem potentials between the 2 coexisting phases, in and out. So in is the inside, phi is a bit bigger, and equal osmotic pressures. And we derived already how they are defined. The osmotic pressure can be expressed, as well as the exchange chem potential, in terms of the free energy densities. Okay? Once you evaluate this constraint, you can calculate a so-called phase diagram, chi as a function of phi, which gives you here a regime where the system is phase separated. And this line here is called the binodal. There's another line, the spinodal. Who has ever heard of spinodal line? Yeah, that's interesting. I will not talk about here how to calculate this in my notes, but come to the colloquium talk tomorrow, because the spinodal line for sure is a line, when you think about it, has no physical meaning. It just marks a linear instability, which is somewhere inside this domain, so where the system is already phase separated. Yeah. However, I will show you it's actually very important for an interesting instability of droplets. So droplets can actually form other kind of shapes. And a shape I will tell you is liquid shell state tomorrow in the colloquium. Yeah? And with this knowledge, I guess you can actually nicely follow the emergence of this new interesting state, which also has been observed in a huge amount of different experiments. Good. I also showed you this C. elegans embryo example where we changed the temperature. And just to make this nice link here, how is chi related to temperature? Essentially, it is related to 1 over the chi parameter, and then when you just ask yourself how this binode looks like, it is just twisted and looks like this, while here then are your phase separated. And in the end, recognize this is the data we had in the experiments of the C. elegans embryo. Good. Any questions about that? Do you want to ask questions? Because then we would, based on this, move on and discuss what is the role of the interface and surface tension to discuss Oswald ripening, and then we are ready to go to discuss more advanced topics far away from equilibrium. Any questions? Yeah. Okay. Okay. Yeah. You decide.
In terms of like A and B that you had defined the lattice with, could you like just help me visualize what an ordered into disordered state would look like?
Sure. Yeah, exactly. So, I don't want to do this. Let's do it here. For example, when I draw a box, okay, of volume V, in the thermodynamic limits of system, when the systems are big, what we actually discuss, we have these 2 domains. I can draw it roundish here just for the sake of illustration. Here we call this phase in and out, but these are phases, yeah. And these phases have different volume fractions, okay? So while this phase in, for example, has and the other volume fraction is out. And what phi was, just as a reminder, phi was phi A. So I can clearly write 1 minus phi B, because this is always true. Yeah? Which means this is an A-rich phase. Maybe let's separate it here. Let's say call this is A-rich. And equivalently, of course, B-poor. And this one is here A-poor and a B-rich. Okay? So this is what it means. That is what we call order in terms of spatial organization of A with respect to B. Does this answer your question? Yeah? Okay, perfect. Who was next? I mean, you also have one. Maybe you have to take a microphone, because then everyone can understand.
Sorry.
Or maybe someone else first.
I had a
question.
follow-up. Like, what does it look like above the critical temperature?
Speak up a little bit.
What does it look like above the critical temperature?
Okay, so where is the critical temperature? Yeah, so the critical temperature sits exactly here. So the critical temperature marks the point, same as here. I can define a critical chi parameter. Yeah. This marks the chi let's talk about the chi parameter, for example. Above the critical chi parameter, phase separation is possible. Okay, this is what that means. Yeah. So also here there's a corresponding critical phi c value. Yeah. So it marks kind of the onset. At this critical point, you have exactly everything you know from classical van der Waals fluid. Yeah, you have critical polysens, etc. These large, long-range correlations, etc. We will not discuss so much the critical point in my lecture. And the reason is very easy, because we want to discuss how Bamberger condensates, or condensates in general, affect chemical processes, etc. And for that, how do they affect this? What is their actual difference? Is exactly the differences between the phases. So for that, we want to be far away from criticality to see something interesting. Now, that's why I will not focus so much on the critical point.
The Maxwell construction that gives the information of the separation phases, because every time that I learned about Maxwell construction, it was just a mean to make the physics right, but in this case, it states that the pi and the mu dash will be the same.
Exactly. So there are two ways of saying the same thing. So first of all, the mathematical algebraic conditions for the Maxwell construction are exactly those things. So they imply the Maxwell construction. And I showed you how to derive those, so we should be fine. If you love mathematics, you go home and you're fine. But if you want to see what this does graphically, yeah, there's another argument. You could say, well, this doesn't work. So the problematic regime is exactly only this one here, okay? This one, everything is fine from the first side. So you could also say, I just connect these kind of two things. You could, yeah? But then you create a kink in the free energy. And then you can ask in the end, what is the only solution where the new free energy is smooth, yeah? and continuous. Yeah? And this is the only graphic construction is exactly the Maxwell construction. So you move down this line, but that's a mathematical argument. There's a bit of physics involved because the free energy density cannot have discontinuities. So it's always smooth, yeah? And it also depends on what? On phi, which is, after the continuum limit, always smooth. So there can't be any kink and any discontinuity on that level. However, derivatives of it can. Yeah? That's another thing. Yeah? So, but the point is, but that's a bit of a physics-inspired math argument. Yeah? And this is simply the way we minimize the free energy, that's physics, and this is, we get mathematical conditions that imply this graphic construction. Yeah? There are different ways of saying the same thing. Yeah? Yeah? Any other questions? Good. Otherwise, I will continue. So, because we have to get to the next level, what we have discussed here are phases that actually do not really correspond to this drawing here. We discussed in the end at the very moment a system where I put the volume to infinity, and the same way I increase this volume of this inner phase together. Yeah? That's called the thermodynamic limit. So these conditions are only true in the thermodynamic limit. However, when you look into a movie, it's obviously a finite size phenomenon. If you see a droplet, it has a curvature, it has a length scale, so these Barber condensates had a length scale of, for example, 1 micrometer. Oh, sorry, it's, oops. Oh, come on, open. And we have to account for this when we look at, in particular, smaller, not infinitely big condensates. And what we have to do, we have to kind of change our ansatz here, or add something, because what we have only accounted for is the free energy of the respective bulk phases of the inside and outside phase. But what we are missing is a contribution of the interface. Okay? And how to do this? I did this already at the end of the last lecture. We can write down, say, I assume it's a spherical interface for a second. Yeah, assuming somehow there's a driving force that if it's not spherical, it brings it back to spherical. This we can discuss as well if you like, but it comes here with an important constant, and this is called the surface tension. Yeah. Okay. And now you see that compared to this previous ansatz, this is a perturbation, yeah? And now we can actually work out the effects of this perturbation on the equilibrium conditions. And as a quick spoiler, you can now repeat the same calculation. So what did we do? We had F as a function of NA and NA in, subscript, NA comma in and V in. Okay, this is what, oh no, we wrote in terms of out, sorry, in the script. But it doesn't matter, you can replace it as you like. And we minimize it with respect to these two variables. So now we just added a new term, and you can do the same thing. Okay, but now it's weird because we have volume and radius, but of course you can express very easily and say, the inside V in is simply a sphere. You can solve it for R and write this in terms of the of V in, and V in you can replace in terms of V out. So you see very nicely, okay, that's the same equation in terms of the dependencies, but it's kind of perturbed. And this perturbation we can actually calculate. And to do this, I will now move away from volume fractions. There will be a reason for that later, because I will do non-equilibrium thermodynamics. and I want to derive what governs fluxes of molecules, and for that I would like to use now the conjugate quantity of the Kemp potential, and this is either the particle number or the corresponding intensive quantity. It's called the concentration and not the volume fraction. But in the end, it's not a big thing because the volume fraction is simply nu times C. Nu was the molecular volume, and C is the concentration, okay? And since nu is constant, it's just a very trivial manipulation, but conceptually, particularly when we do later Onsager linear response, it makes a difference. Good. So now we write everything in terms of C, so all the phi's get replaced by C's. That's not so hard. But what we can do now is I can define now here an object, a new object, in and out respectively, and I call this EQ for equilibrium concentration. That's now the equilibrium concentration with this extra with extra contribution of the surface tension. Yeah? And I write this in terms of what I had already. C in, C out. So this one is the same thing. This one is simply phi in, out, 0 is C in. Oh, well, I can write like this. Nu. Just divided by nu. Okay. Plus a perturbation, and which I call delta C in, out. Okay. So you just plug in this in because I would like to not just solve this new condition with the minimization, but I would like to get this extra contribution that comes, like to separate it from the surface tension effect. Okay? I mean, that's the same calculation. So I don't want to do it because it's essentially algebra, and you can solve for it. And I do a linearization. That's why I wrote also here with the delta. Linearization essentially means, in a nutshell, that any delta square term gets neglected. Okay, that's what it means. So I get naturally, because the Kemp potential and the osmotic pressure, these are non-linear functions of the volume fraction and thus also of the concentrations. But if I linearize, we'll discard these contributions. Good. So what do I get? We get a very famous relationship, which we will need in all talks. Yeah, so I exactly write this equation now for respectively the inside and the outside phase. It will depend now on the radius. And we know how it must look the same way that if the radius goes to infinity, I must get back this one. Why? Because what I added here, this one scales with r to the cubic, and this one with the r to the squared. So for r to infinity, they are always dominant compared to this one. Simply volume to surface. Please, yeah.
Well, it's not very clear for me. Why did you connect the density phi with the surface tension again? Can you repeat? The phi equals to gamma c there.
Yeah, so before we calculated, without this one, we calculated the equilibrium concentrations, but without the effect of surface tension. Okay. Now I add a term of surface tension. Now they somehow get modified. Yeah, because of this term. Thus, this modification is exactly this one here. This is this perturbation here. Yeah, will now depend on surface tension. Yeah, that's exactly the reason. Yeah? Because you just solved the same thing and you added something, then of course, then you get a difference. Yeah? Okay, and you can nicely rewrite this in terms of the old solution. And you get this. This has a very famous name. It's called the Gibbs-Thompson relationships. When you do the minimization, maybe this is exactly what your question was asking, because I didn't write actually how they are affected. When you calculate now these perturbed, when you ask yourself how the equilibrium conditions are affected by surface tension, the equivalent of the exchange current potentials are not affected. But what is affected is the osmotic pressure balance. It gets exactly 2 gamma over R. Yeah? And this has also a very famous name. This is called the Laplace pressure. Yeah? So we see these new solutions graphically still must have the same slope on the free energy, but they're upshifted. Okay? I have to tell you what this L is. It's a length scale. It's positive. I define in a second. This is positive. So you see that both concentrations are upshifted, so 1 plus, compared to the case without surface tension. So if I find a second color here, yeah, maybe. Let's try that one. So what I get here, I find 2 new solutions where, let's say here, which are still parallel, right? Okay, so still parallel because the slope is still the same, but they're both upshifted to the right because of the effect of Laplace pressure. So when you have a very tiny droplet, this effect can be quite significant, which means that the actual measured concentration inside and outside is uplifted by the effect of curvature, by the effect of surface tension, and it scales with 1 over r, so small r means big changes. However, when of course r is much, much bigger than this length scale, then this is just almost 1, this bracket, and the effects are negligible. So it's very important to think about how big is this length scale and what it is. Yeah? So this length scale has a name. I'm very sorry, it has a very confusing name, because it has a name that is already taken by something else. It's called the capillary length scale. And I'm saying this because historically it was a bit unlucky. People have made up 2 capillary length scales. So there's one that is hooked up with gravitation. This is not what I'm talking about. No? There's important essentially when droplets deform because of gravitations. When this is not what this length scale is. This is also called capillary length scale. This exactly tells you how much curvature can affect the concentration inside and outside of a droplet. And we can calculate it exactly because we have now, by putting this in, we obtain expressions in terms of the free energy. I can actually write what one gets. Of course, this length scale depends on the surface tension. It also depends on how strongly you phase separate in your phase diagram. It depends on your curvature. So this is the capillary length scale outside that you obtain from the calculation, and you can also calculate it for the inside. And this here I express in multiples of the capillary length scale outside, the inside one. Okay, so we see something very interesting that people like Lüschwitz, Slusov have exploited in their seminal papers on Oswald ripening. And this is simply that L gamma in is typically much smaller than L gamma out. How can we see this? Well, here I already rewrote L gamma in with respect to L gamma out. So what do we have here? We have, first of all, a fraction of the free energy curvatures When we look back, essentially what we look at is here is this local curvature here and here. Okay. Yeah, it might of course be different, but actually typically they are rather similar. Yeah. Yeah. So this is order 1. However, we have here a very interesting fraction. This is C out over C in. This tells you how strong is your phase separation. So in terms of this plot, how far you're away from the critical point. When you're very far away from critical point, the fraction of the inside to the outside gets extremely big. And that depends where you are. In cells, this is often between 10 and 100. In vitro, it's often in the order of 100 to 10,000. So it can be a very big number. And we call this strong phase separation. Which simply means that C in 0 is much bigger than C out 0. And then you can verify that actually I can essentially neglect this term for practical purposes, assume that the inside concentration is almost the one I get from the classical Maxwell construction for infinitely big drops. However, for outside, I have to account for this effect. I want to still mention, I don't do this calculation, we can now express, of course, if you want, we can calculate and estimate this number, okay, with a real free energy. I mean, these are all things we can take from real experiments. We know surface tensions of condensates. On my slides, I might have not listed surface tensions. I just realized only viscosities. But we can calculate it. And of course, it has to be taken with a grain of salt because it might not be true for all systems, but at least all the systems I've seen in the last 10, 15 years. So I write here typically. Yeah. This is microscopic. So what is here the microscopic length scale? Yeah, this is simply the molecular volume I have to the power 1 over 3. So it's in the order of the molecule size. Say you have a protein, it's a nanometer big. This length scale is a few nanometers typically, which means that the change here of the concentration here is a few nanometers, and if the droplet is 10 nanometers, it's significant, but if it's a micrometer, it's very little, okay? Now you might ask, okay, well, you discard already this one, and you now tell us this one is super little. Why the heck are we doing this? This little difference still drives a phenomenon called Oswald ripening. That's why I have to discuss it. And it drives coarsening of droplets. And when I have many droplets, remember the video I showed you with many droplets, that on long time scales, they end up as a single one. And that requires exchange of molecules. And this relies exactly on this little difference here of this little upshift. Okay, that's why it's important. Good. So the upshift I discussed, this is perfect. Good. Now it's ready to introduce space. Because in some way, I still have separate space in these two bulk phases. Now we added here the interface, but I just was drawing a line. But when you would go through this interface, you would expect, well, this is not just going from one phase to the other through a mathematical line. Actually, there should be a smooth interface. Yeah? And this I would like to calculate now. This is part 5, the interface profile. So we have to minimize, again, the free energy. But now we minimize the free energy that we actually derived. So here, remember, in my summary, I only wrote the density, but we obtained more. We also obtained a gradient term. But once I have this gradient terms, I write the free energy, and I use here now these square brackets to indicate the fact that this is a functional, and it depends on C and the gradient of C. I can write it as a spatial integral of the system of the density plus our gradient term. We introduced constant kappa this was actually our free energy. Yeah, and I kind of, without telling you so much, I kind of did not talk about that one. Yeah? Well, it's a gradient term. It only is relevant at the interface, and we somehow made our way from the thermodynamic limit down to the first interface contribution, but now it's time to minimize that. What do I have to now? What is different now when I minimize this? Before, we just calculated derivative with respect to the two variables. What's now different? What I do now? So, what is now different here? What is the type of derivative code I have to use here now? How to calculate a derivative of a functional?
As I see it, it looks like there's only one variable now.
No, two.
Just the two.
Two,
The C and
but that's just
the
right, yeah,
the derivative and the divergence of C.
And yeah, but what is the type of? Yeah, it's not a function, but it's an integral. So what is the derivative I have to use? Have you seen that?
I don't know. It's just the term inside the integral.
Yeah, but okay. So this is a functional. So the derivative that I have to use now is a functional derivative. It's typically written like this. Of f. Okay. I made a little bit how to. No, okay, I did not make notes on this. And this functional derivative of this function, I do not just minimize it. Because if I minimize it, I get many solutions with different averages of the concentration. And what I would like to maintain, a constraint. And this constraint is, I call C bar. It's integral of C of x. What do I have here? Oh yeah, okay, you can write here V, 1 over V, okay? So this is, what is this? This is the amount of particles in my system, okay? This I would like to keep fixed in the minimization, yeah? What do I have to do when I minimize with a constraint? What's the method to minimize with a constraint?
Derive equal to 0.
Second?
Derive it and equal it to 0.
Perfect. But there's a constraint now. So it's, you're not just performing, do not just minimize it, but with a constraint.
Using Lagrange multipliers.
Perfect. Exactly. Yeah. You introduce a Lagrange multiplier. Yeah. Exactly here for this constraint. Yeah. So we can write it. Where lambda here is the Lagrange multiplier. Good. This gives us, so what do we get here? We get essentially the functional derivative just kills this one, and the integral is just I'm left with a lambda. This is just a constant, right? This is our constraint. And now we get a new definition, a generalized definition of our chemical potential, which is simply the functional derivative of f. Okay? When you calculate it, you get the definition that we had already. It's the same as here one. We had here, look at this one, we had derivative of free energy with respect to the volume fraction. I can put the volume fraction in here and just use that the concentration is the volume fraction divided by nu. Nu is a constant because it's incompressible, so it's simply I can just take this in and write C here, okay? And the other term I get is because of the derivative, I lose the 1 over 2. That's the reason why people write it like that. And I get here Laplacian of C. Oops. This is my function derivative, okay? And now we see that this chemical potential is now no more an easy function of C, it's actually now a differential equation, because I have here Laplace of C. But it is, this is 0, yeah? It is lambda, and this is constant. Okay? And now I can solve this differential equation. Okay? And this is a very difficult differential equation, in particular when you use this Flory-Huggins free energy. The mathematical reasons are the logarithms. They are really difficult to solve. When you open up a textbook, I mean, you typically don't find this because people focus on critical points and they already hang around with Ginzburg-Landau and have done a critical expansion around the critical point. Then actually this object gets much simpler, yeah? And also then it's rather easy to solve. Yeah? And the solution you get, so is a very famous solution, and this is C of x, C out EQ, it's a different board, now realize. Have been replaced. So the fundamental solution, so these just take care of the boundary conditions, that the integral is exactly conserves this C bar, yeah? Is a tan-H solution. This is the famous classical interface solution. However, you could ask, yeah, but well, the critical expansion you didn't do now, yeah, how to do this, etc. I would assume that you have seen this in your courses, and it's just expanding this around this entire thing around phi, where is it? Phi minus phi c here. And then you have to go up to the fourth order, you get a fourth order polynomial, and can obtain that solution. When you compare the actual numerical solution, for example, of this one with this tan-H solution, I can tell you, if you just do the plot, you will not see a difference. If you plot the residual, there is a little difference. I went to the literature, I found only, we did then the solution in 1D, in 3D I've never found a solution to the interface problem. I think I don't want to motivate you to do it because the residual is really little, but it's not done up to now. Maybe it's even not possible. But to obtain it, so it has always this shape. Yeah? So what you should remember, how this solution looks like, it looks like this. So there's a position of the interface that is fixed by this constraint. The inside concentration is this, and this is here the outside concentration. C out EQ. This is C of x. Yeah. And exactly this domain here is exactly called the interface. Okay. And that's how the spatial problem looks like. However, when you do the critical expansion from the Flory-Huggins, that came actually from a lattice model where the chi, where the kappa, so here, where's the kappa here? Here. Where the kappa actually was depending on chi. Yeah? What you can do now, you can express this interfacial width in terms of the microscopic parameters. And when you do this, you find exactly what we expect. The interfacial width, the length scale of the interfacial width is exactly set, first of all, by the size of the molecules. Yeah? So it's really almost microscopic length scale. However, it gets multiplied by we have only one parameter left. This is chi. Yeah. So this is here the interfacial width. So it increases with chi, it's just a square root dependent somehow, right? So chi, what is chi? Chi came from the e's. The e's were interaction parameters. Well, to keep a thermodynamic problem, they are in the order of a few kBt, okay? So now I have chi over chi minus 2, so if they both plug in something, 4 kBt, I don't care, something like that, right? Not 4 kBt, number of 4, because they measure the multiples of kBt. Then you have 4 divided by 4 minus 2, okay, gives a 2, the square root of it, okay, not a big thing. It's very difficult to create an interface that is far above this molecular length scale, okay? So when I draw molecules here, proteins, then they would look like this, likely, okay? That's how you should picture it. Except, you see this? There's a divergence here. When chi goes to 2. Chi goes to 2 is weird because I did this here already for symmetric. The critical chi is 2 for the case n being 1. For n getting bigger, this just shrinks. Okay, then just there's an, I didn't calculate it for the n because it's very complicated. But this is here, this is a result for n being 1. No? So you see at the critical point it diverges, and then you get a very broad interface. And this is consistent because criticality tells us that you can't distinguish between the two faces. The corresponding order parameter is phi, is phi here, that's the order parameter, and it becomes, the faces become indistinguishable, the values become the same, and the connecting domain is now the interface, so it has exactly to smoothen over the system and diverge. That's kind of makes completely sense. Just for the people interested in criticality, this is how you see it. However, we stay away from that. So what you should, for the remaining of the talks, recognize is the interfacial width is typically set by a few molecule sizes. Okay. Good. Questions on this? So I think now we can close the introduction part on thermodynamics of phase separation. It's time to talk about dynamics, because actually this is exactly what I would like to push you a little bit to think about modern problems away from equilibrium, about phase separation in cells, in presence of biochemical reactions, leading to all various patterns and exciting physical phenomena. And that's why we have to now discuss, do a first step towards the dynamics of phase separation. Good. Of course, I could do it super general, if you like. So you think right away that we could generalize all these calculations. We can include here an n. We can go to a 10-component mixture. There are lots of angles you can think of, but of course, then it's just the calculations just get longer. But conceptually, they are on the same level. That's why I always consider mixtures with the number of components that is relevant, that makes the point. But of course, we can look also at more. So when we only talk about phase separation without chemical reactions, I look just at it as an AB mixture as we did it up to now. Each component is conserved. So when you integrate over all the A of the system, the total amount of A is fixed, same to B. When you do this, and I want to write an equation for CB, I know that the integral, so this is now, I want to now write dynamic equation as a function of x and t. If I know it's conserved, what can I immediately write without even thinking further? Yeah? You want to lift your hand? Yeah?
If you integrate over space, you get 0.
Exactly. But what can I write here on the right-hand side? Not 0. No? There can be a contribution. What's the only contribution I can write?
Oh, the boundary?
Yes, exactly. have a local volume element, right? I ask in the end locally, how does the concentration change here? It can only change by
The boundary of that region, the neighboring cell?
Exactly. It can only change by boundary fluxes. So what mathematical structure do I have to write here on the right-hand side?
A line integral?
Yeah, and I can convert it into, it's a local equation, it's a local differential equation.
A space derivative?
Exactly. So into a divergence, yeah? Can always write here divergence, yeah? Nabla dot of a flux. That's the 3.5. Yeah? So this means that the concentration locally can only change by the in or efflux through the boundary of the volume element. Yeah? Also, you can also call this continuity equation if you like, etc. Yeah? Of course, later when we do chemical reactions, we would add here local sink and source terms. Yeah? That's what we do. We then will break this conservation law. Okay. We talk about an incompressible system here. I already mentioned what is incompressibility. That the molecular volumes are constant. I can thus write that the volume is just composed of these two particles. This is clear because this I introduced the lattice. You hear with the lattice size m. I just multiplied by this volume of a lattice side. Okay, you see this here. This is very nice, very self-consistent. And now I can write this in terms of concentrations. Yeah? So this is a constant. Yeah? You see like, this is almost a concentration. Yeah? This implies right away that when I write this in terms of concentrations, that dTA, yeah, is minus dTCB. Except, be careful, yeah? We have to add here this kind of fraction. Yeah? It would have been easier, by the way, as an aside, to make a point of why people love writing this in terms of volume fractions. I can also write volume fraction fields. Yeah, so the volume fraction field here is, as usual, defined like this. Okay. If I would have written this in terms of volume fraction field, I don't have to even think. Yeah. And you can see right away when you rewrite this in terms of C, you get that equation. Yeah? So it just reflects in a way that if I take an A molecule out of this local volume element, and if it has not the same volume as the B molecules, because we only can have exchanges, I have to make sure that the same amount of volume is exchanged. Yeah? Otherwise, the system undergoes compressions, and this is inconsistent with incompressibility. Good. So, time to write now the flux. The flux can have two contributions. One is simply advection by some velocity field, say hydromic flow, for example. And we define here the exchange current. Yeah. And when I want to write the flux JB, I can also have an advection of B. Yeah, but the problem is the exchange current need to have opposite sign here. And again, I have to be careful here and add exactly this nu A over nu B fraction here. Otherwise, it would be inconsistent with that relationship. And now I think I will clean at least a little bit the board here on the left-hand side. I think I need much more space then, if I could get your help, Ricardo, at some point. But I just cleaned this one because this was a summary to continue with the dynamics. Yeah, they're just like everything until the dynamics. Yeah, so leave the dynamics, that would be great. Yeah, thank you so much, Ricardo. I feel a bit bad to ask you for that. So. But I hope it's better readable now because I write much bigger. Is my handwriting now? Okay, cool. Because
it's a lot of space in the first rows. That's also a good way to see small.
But otherwise, as I said, if you can't read something, then please let me know. Good. yeah, and we got one condition, of course, not to violate incompressibility. I also need to have that the flow is incompressible. Yeah. So now we could continue, of course, with flow. Flow means we have to think about the coupling between molecular transport via diffusion of molecules and the hydrodynamic flow. So this will essentially lead to a coupling between the Navier-Stokes, with an extended version of the Navier-Stokes equation, with the corresponding transport equations, which we will call later Kahn-Hilliard equation. If you're interested in that, I'm happy to do this for you. This is really a nice calculation. But today, let's keep it a bit simpler and go into the reference frame. Where the velocity is 0, okay? So we don't have to think about the Navier-Stokes type problem, but we can do it. We can always solve the same problems also with flow, yeah? And it's relevant because think about two droplets are next to each other, they touch each other, they fuse. So when they fuse, there's net mass motion, and there will be flow. Yeah? So generally, it's just there. So good. So this equips us with a very simple equation. Where now rho CA is equal, we just call it like C, just to avoid this extra subscript A. This is a very simple equation, but it's thoroughly defined because the exchange flux is defined to a net mass motion of the fluid. We just put it to 0 to keep things simple, but it's a well-defined exchange current. So the next question is, what is the flux? Okay, how can I write the flux and link it to the thermodynamics we have discussed up to now? And the general recipe it's called irreversible thermodynamics. That is the classical way to link local thermodynamics close to equilibrium to transport. I have in my notes, I made a box which I call irreversible thermodynamics in a nutshell. So in case you've never seen that at all, honestly, it will be very difficult to say, oh, of course I understand now this step. So I give you some intuitive arguments. You've never seen it. If you want to learn it, look at this box. It gives you a quick teaser. But in general, irreversible thermodynamics are the calculations where you have a page and have to turn it like that because the calculations get so long. And they're really boring in a way because they follow always the same recipe, but they're very important to do for different systems. But let me tell you what is the basic idea. The basic idea is essentially it's grounded in the second law of thermodynamics. Which just says that the entropy production, the total entropy of the system is either positive if you're not at equilibrium, and it is 0 if you're at equilibrium. Okay? That's the basic idea. Yeah? Now, to use this, we have to link somehow this total free energy, this total entropy to our free energy density that we have already. Yeah? And this one can actually do. Yeah? So like one can multiply it by a T, that's good, because then T times S is an energy, right? And one can link it to maybe I can write this here. One can actually show, yeah? Wait a second, it's minus when I can actually show that the total change of the entropy times T is minus, so this is a spatial integral of the time change of the free energy. There are boundary terms. That give in the end we can neglect. For the experts in the room, they give the non-dissipated fluxes. We don't care about at the very moment. And when you do this calculation, and in a nutshell, if you do it with hydrodynamics, if you do it with charge transport, if you do it with other kind of outcome effects, it's always the same idea. What you have to do is you have to evaluate this calculation here. Yeah, you have to apply this time derivative on phi in this one. It will give you back the chemical potential, and you have to use all the transport laws, right? And the transport laws we have already is, oh, I wrote phi. Just sorry, I didn't want to confuse you guys. You can write C, of course. Yeah? That is the transport law you have to use. Yeah? When you have hydrodynamics, you also use relationships called like Gibbs to Hem relationships, but we don't do hydrodynamics today. Good. So if you do this, you get a very beautiful result. And what can you expect? When you do this, we have to somehow, in the derivative, there's a derivative of f, so this gives us the chemical potential, yeah? And we have the flux that we obtain. And after a little bit of partial integration, we get a product, scalar product, of this flux J with the gradient of the exchange chemical potential, okay? So this contributes to the dissipation, this product J times gradient of mu bar. And how to make this positive? Well, here's already a negative sign. To make this positive, I can come up, and this is called linear, goes back to Onsager response. If I write the flux minus, to kill this minus here, times a positive coefficient that can depend on C, times gradient of mu bar, you see right away that this is actually fulfilled. Because then I get here, the minus gets killed, and I get a gradient of mu bar squared. That is always positive. The spatial integral of this positive thing is always positive. Then it's fulfilled. And it's called linear Onsager because, I make it a bit too much here, it's the first order. So you might anticipate, if you've never seen that, we could also include a second order or a third order. No worries, you can do. But the first order is non-negligible, and this is definitely what we have to minimally account for. So you can see this also as an expansion and go to higher orders if you want. Okay, but then you get higher orders in gradient of mu, so you need large gradients in mu bar that they're relevant. Yeah? What is this coefficient here? This is an Onsager response coefficient, but let's make it simple. In that case, we would just call it a mobility. So it tells you how much flux you obtain given the gradient of this exchange chemical potential. In irreversible thermodynamics, this is also called the flux, and this is also called the thermodynamic force. So in the end, this gives you a flux-force relationship, and you just have to add a positive coefficient that tells you how fast things are transported. And we will see then later that this one multiplied by kBt will give us, talk about molecular transport, what tells you how fast molecules diffuse. What is the coefficient that tells you how fast this happens? Yeah? The diffusion coefficient, exactly. So it really will be the definition of the diffusion coefficient, but it will be a collective diffusion coefficient, and I'll tell you in a second what that is. Good. So we can combine this now to one equation, and this equation has a famous name. So it gets a plus. We have to keep here mobility inside. Oh. This is called the Cahn-Hilliard equation. Sometimes people call the expanded version of the free energy to calculate this one as candidate equation that comes from a Ginzburg-Landau equation. But since it's a special version of this, this is just a generalized version. Yeah? So this is belongs to the class of candidate equations. And the mu bar here, I wrote already. Is it still on the right-hand side? No. Just to have a complete set of equations, a simple functional derivative, which is which can be calculated like this. Okay. So which means now you have the Floer-Huggins free energy. You can now plug this in, calculate this. You even know how this depends on kappa, and you get a beautiful partial differential equation. You can solve on the computer or treat further analytically. Yeah? This equation is a very complicated equation, and because it's nonlinear and it's fourth order ingredient equation. How can you see this? This is here gradient. Gradient gives a Laplace of this one. Okay, this is the Laplace squared equation. And that's why this is in general a complicated equation. Moreover, to be fair, if you're able to solve it, then you push the button and you see a nice movie, say of a droplet doing something interesting, and you will see lots of movies in the next talks. So if you're like, oh, come on, show me some movie, it will be extremely many movies the next days. Yeah. But we often solve that equation. But there's a little bit of a problem solving that equation, because then you have this movie, and what's then? It's a bit like having an MD, molecular dynamics simulation, and you have a molecule doing something like this, and then? What do you do then? I mean, you have to understand what's going on, which means in the end, this equation, it's just the starting point for any analytic further approximation to really understand underlying mechanisms theoretically. And this we will tackle now step by step throughout this and the remaining lectures. Good. So what number am I? 7. On the first step is narrow interface model. I show you now the most easiest version, and we will exploit this later in many other projects. Narrow interface model. I mean, okay, what do we have to? What are the problems here? Yeah, simply want to get rid of both the nonlinearity and the higher order gradient terms. Okay, so when I draw a profile, how would this typically look like? Well, you can have some gradients inside here, la la. Okay, wait, okay, I said here's some gradient, I don't know. Let's say this is our C of x. And why can I draw it like this at a given time t? Because I know that the system has a strong tendency to create this interface. And even though there's some out-of-equilibrium dynamics, there are fluctuations or whatever, if it were not a droplet, if these concentrations were not close to the equilibrium concentration inside, or how did I call this? 1 second. No, the other way around. I have to always remind myself about the notation I'm using. Okay, otherwise we will not cannot talk about a droplet at all. If you say like, no, fine, I have such a crazy out-of-equilibrium situation that my profile looks all over the place, I was like, yeah, then we should not solve that equation. Then we can come up with a different equation. Huh? So this is a bit the kind of the reference state, and then of course you can think about, okay, we could maybe expand inside and outside this Canhillet equation. And this is called narrow interface model because we want to get rid, first of all, about the smallest length scale. This is, by the way, I should maybe mention this here. This fourth order term that creates the interface. Yeah? Remember our calculation. The kappa term was depending on chi and gave us exactly this interfacial length scale. So it creates the interface. And I mentioned to you, when you have a real problem, you have some molecule sizes, say a nanometer protein, then the interface is as the smallest length scale of the problem, of a few nanometers, and you want to tackle a problem that lives on a micrometer scale, for example. I mean, okay, this is numerically no way to bridge. So It's a big problem to even use realistic numbers in a Cahn-Hilliard equation that corresponds to real experiments. So the smartest way to do this is to find a systematic way to integrate out this problem, because the interfacial length scale carries essentially one important parameter, and this is the surface tension. This you want to model correctly, but whether it is now 1 nanometers or 5 nanometers should not be a big problem. Should not be a big issue. So you want to kind of remove this one here. So you would like to assume that the actual profile here makes a jump. This is called narrow interface model. So remove, first of all, the smallest length scale of your problem to make the system tractable. Plus, you also linearize in each phase. And the way we linearize, we will do it differently throughout the different talks. But for the classical, just an classical emulsion in a box, we use what we have learned up to now. We just say that the inside EQ is approximately C in 0, so it's just constant. Yeah. And outside, we expand C out equilibrium plus delta C. Yeah. We just write it like this, okay? And then linearize our equation. So that's how we remove exactly these non-linearities here in the chemical potential. And that we do not have a crazy notation now, I just call this C, because otherwise I always have to write Laplace of delta C, and we forget what the delta was. Yeah? And you can always calculate it back with just adding this offset. So what do we get? So we get rid of the interface. So this thing is out. And now we linearize this thing. So it just becomes a constant. So what's this thing here? The gradient of mu bar gives you then essentially, without this term, gives you a gradient of C times a constant, right? When I linearize. This is what you get. Well, the constant I can take out, right, with the Laplace here. What do I get? I get the most simple equation we all love and we all can solve pretty well. We get a diffusion equation for the outside phase. So the associated problem then looks like this. It doesn't mean that outside it's flat, because outside I have still to make sure that here I have the boundary condition right outside the droplet. That's what the interface creates. And far away, I get some sort of concentration here. And if I make a snapshot of the dynamics, the situation might look like this. But the outside field here is just governed here by a diffusion equation. And this drawing, you see like how the drawing looks like, yeah? Well, the drawing, what does this equation also tell us? That the flux here then gives us D is a constant, the diffusion constant, gradient of C, and this is called Fick's law. So essentially, by linearization outside, the flux, we also get a relationship how to calculate the flux. Then the flux now is no more the gradient is proportional to minus gradient of the chemical potential. So molecules move down the chemical potential gradient. Now we get the classics of that the flux is proportional to the gradient of C. And in that case, we can look at the slope here. It would mean that the droplet sucks up material and the interface would move, for example, in this drawing. It can also be, I can also draw another kind of situation if you like. And I've not defined the diffusivity up to now. Let us maybe write it here. Then everything fits nicely together. in this linearization, the diffusivity is simply a product of the mobility times what you get from the leftover from the thermodynamics is the second derivative of the free energy. Okay. Maybe this is. I have to maybe write here C out 0. Okay. And the mobility is positive, and it gets multiplied by the curvature. Well, is the curvature also positive? Of course it is. Remember that the free energy after the constructions looked like this. We talked about these points here. You see when I evaluate the curvature, at this point you see it's positive. Also this point is positive. So both are positive. Yeah, this is positive. This is positive. So we get a real diffusion coefficient now because diffusivities need to be strictly positive, of course. Good. That's an air interface model. And why so powerful? Because we can solve it. And this allows us to discuss growth of droplets. And I first will discuss a single droplet because of simplicity. So what is growth? Growth is simply movement of the interface, or shrinkage, yeah? I mean, left or right movement. And we have to define some interface velocity for that. Which I call UN is equal to, and N is the normal of the interface. So I have to do a little bit of geometry. We keep it spherical today, so it's not so difficult. So this is the center of the droplet. Here is the interface. This distance is called the radius R. And this point here is exactly this interface point we would like to calculate. And this N here is the normal vector. Okay. Good. So when does an interface grow? So what do we have actually? We can I can draw the interface here again, and then draw here two boxes, which you can understand as discrete volume elements, right inside and outside of the interface. So this is the interface again. And I call this here in, this out. And an interface moves if I have a flux difference between these two volume elements, right? That's an absolute prerequisite. This is called the growth conditions. So that the flux J in, this is here, is not the flux J out. So if they balance, nothing moves, right? It remains steady. It's just a prerequisite, right? So first we have to define what it is. The flux right inside, So this is just a construction. So in idea, we then do a continuum limit and make these volume elements smaller, 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 that they collapse exactly at this line. So with this, we can write here that we define here interface velocity right inside. So here. And what do you convect? You convect C in EQ and a flux outside, and you convect outside EQ. So our flux is always the velocity times what gets moved. And what gets moved is a concentration in that volume element. Perfect. But this can lead to problems, in particular when these velocities are not the same, because then the interface is no more well defined, because what does that mean if they don't move with the same speed? And this is called interface integrity condition. And this integrity condition just says that that the velocity u in is out, and this now I define, or that is exactly this interface velocity I defined here. Okay, good. This allows me to rewrite this in terms of un. Let me see. Yep, I multiply by the normal vector, and then I get simply that the local interface velocity here, or here, is given by the flux difference projected on the normal vector divided by the concentration difference. And this is the interface movement condition. Good. How to calculate these fluxes? We need a thorough mathematical condition. We can just write this as the flux. I mean, the flux, first of all, we can evaluate everywhere inside and outside by the Fick's law. We can evaluate this Fick's law exactly at this position, R. Yeah, but now we have to move a little bit left and right, right? Yeah, so I move a little distance A, left and right along the normal. And of course, to have a proper definition, I put A to 0. Yeah. So essentially what you do is, because you can evaluate the flux everywhere here, and I want to get exactly flux right inside the interface and right outside the interface. Yeah, that's how it's meant to be. And yeah, then we're almost done. Special case for today is spherical drops. And so the normal vector is simply the radial unit vector in radial symmetric coordinates, and our Laplace in our transport equation is simply 1 over r squared dr r squared dr. Okay? So we have to solve, to get the flux, what equation do we have to solve? We have to solve that equation. Okay? This everyone can solve, but everyone who has solved a boundary value problem of a time-dependent diffusion equation still feels like, oh, is this worth the time? No. And there's a trick. So then start enjoying the calculations, and this is called the quasi-static approximation. It just says the time change of the profile, okay, I have to be careful, can be approximately neglected. Okay? This does not mean the system is steady. That's why it's called quasi-static solution. Okay? It just means that, say, this is the interface. Molecules diffuse, the profile changes, and then the interface starts moving. And on the time scale the interface moves, I always see a quasi-static profile. So I don't see the transient of the diffusion equation. Okay? This is what means quasi-static. So it's not stationary. It's just on the time scale of the interface movement that the time dependence of the profile can be neglected. Whether this is always fulfilled or not for these problems is not so clear. Yeah? So that the mathematical condition is simply, on the time scale, you move slow enough such that you're always quasi-static. Yeah? But you feel like it can't be true if I'm a single droplet in an almost infinitely big space, because it takes me infinite amount of time to diffuse at the very back of the room, right? I mean, that will not work. And we would discuss later emulsions, and emulsions are simply, I better go inside the room. You also are drops. I'm a droplet, and the only thing what matters is how long it takes to diffuse from me to my next neighbors. And on that time scale of my growth, the profiles between us need to be quasi-static. and the same between you and your neighbors, etc. And that is pretty much well fulfilled. And actually later, when you do the full calculations, you will see that the growth of droplets is very slow. It goes with a power law with an exponent 1/3, and diffusion has 1/2, so this is completely safe. But yeah, one could do it and later check for consistency if one is not sure. Good. We can solve this with these boundary conditions. I think the boundary conditions I already have drawn here already, so we can solve this with this boundary condition here inside constant, and far away from the droplet, I call this now C infinity. Okay? This is a well-posed problem similar to electrostatics, because what I have to solve now is a Laplace equation. And we all can solve in all kinds of coordinates since the electrodynamics course Laplace equations. Yeah? But it will be a time-dependent boundary value problem. But let's first of all solve it. So for the single droplet, of course, stationary, and we get C of R as a solution, C infinity plus okay, so you get all these solutions. The logarithm of R is immediately out. You get all these solutions, R to the alpha and R to the minus alpha. And by doing these ansatz, I assume that you have seen these kind of things in your lectures, you get that solution. You can also just plug it in and just prove me right or wrong. So what do you do now with this profile? You just use now this flux equation to calculate the flux. And what you get is this result. Oh. Okay, so you can calculate the flux, and you can calculate the flux at the interface, if you like, yeah? Because you want to calculate J out. J in is 0 because we approximate this problem with a constant equilibrium concentration inside. Okay? If you say like, oh, why not solving it inside? There's a very simple argument. Oswald ripening, or later, the ripening of droplets has the following role. In the end, most of you get extinct in time. Yeah? So, which means there are less and less droplets. Which means, but I'm growing, but very slowly, but the distance between me and other droplets is increasing. Yeah? So which means the time to equilibrate any concentrations on the length scale of my size is easily fulfilled. So I'm easily homogeneous, while there will be continuous gradients always between different droplets. So the system is somehow has a very natural dynamics to fulfill that this approximation remains true. Good. Yeah, and then I think we can finish and say, yeah, okay, that the interface velocity, un, in spherical coordinates is simply, so if you have isotropic domain growth, is simply the change of the radius. And then I can derive a very beautiful equation for the radius, which includes the diffusion constant, a fraction of the equilibrium concentrations, here 1/r, and then here an epsilon minus L gamma. L gamma is simply my L gamma. I don't know, they have a subscript, I suppose, out, over R. So this is exactly this Laplace pressure. And to derive this one, I simply have used this limit of strong phase separation where the inside is much bigger than the outside. Okay, so this is quite common approximation, in particular for the problems I'm interested in that are non-critical, because here, have a look at here, this one here in particular, you have here something big minus something small, you just neglect the something small. And then you get this very famous equation, which you find, for example, in the paper of Lifshitz and Sliusov. This gives you a growth law for the radius, which we can draw here. So we can plot this as a function of the radius. We can introduce a so-called critical radius. It has nothing to do with critical phenomena. Critical radius, which is simply defined at this capillary length scale divided by epsilon. Epsilon I forgot to define on the right-hand side. It's C infinity divided by our equilibrium concentration minus 1. It has a very famous name. It's called the supersaturation. Supersaturation. This is simply the excess concentration available to phase separate. So if someone says like, yeah, the room is, the air is supersaturated, then there are some water molecules who could form liquid droplets. If it's unsaturated, they can't. Yeah? So this is simply the concentration, or the difference, if you like, to phase separation. That's available for phase separation. Good. And then you can make this plot. So that here's here a critical radius. And when you look at this one, how does it look like? Yeah, it has these 1 over r, 1 over r. So for large r, it's positive and decays. It looks like this. It has a fixed point where exactly the fraction vanishes, so where epsilon is L gamma over r. This is exactly the definition of the critical radius. It looks like this. Okay. Here it goes to 0. Sorry, just for clarity. Okay. So it tells you. So what does it do first of all? Here it's negative. Here's a 0. Droplets shrink here. And here's growth. You see the growth here is positive to the right. So if I sit here, I move to the right. So this is called a fixed point. Is it a stable or unstable fixed point? Louder, just unstable fixed point, exactly. So which you typically mark here with this open symbol, unstable fixed point. And we can add here, if you like, these kind of things. So if I'm below the critical point, droplets shrink. From above, they will grow, and they just grow unboundedly. Exactly. So, last thing, and then we're done. This allows us to have a very simple-minded approach to an emulsion. An approach you could put in your computer right after the lecture, if you like. And we have solved now a single droplet. So now we construct a box of many of those droplets, and we consider that they are far apart. When they are far apart, is they do not see really the other droplet, but what they see is some sort of shared field. Yeah, so this one sees this shared field, and this one sees a shared field, and that's how they are get coupled. And this shared field is C infinity that depends on time. Okay? Because if a droplet shrinks, puts here some molecules, the amount of molecules in the outer phase plus the molecules all inside these droplets need to be conserved. So we're not eating up any material. So if they are far apart, we can just assume, okay, this behaves like a single droplet seeing exactly this epsilon. Epsilon was here, or here I wrote the epsilon here. So this was first of all just a fixed parameter, but now it becomes a field that depends on time for many droplets. Which means we can now write coupled equations, first of all with an algebraic one for many droplets, where we sum now over all the droplets. Let's keep them spherical. We have n droplets. The number might change in time. So this is the total amount of material inside n droplets. Then let us look at how many molecules are outside. I just have to multiply C infinity times the volume outside, and well, this correction is not so relevant, but for consistency, we can keep it, and for clarity, we just subtract this little volume occupied by the droplets. Yeah? So you see, this is simply a mass balance equation, but it leads to a coupling. That leads to a coupling between C infinity and the radius that depends on time. And we can combine this. As you have seen here, I added here an extra subscript I with this radius law. I just rewrite it. It's the same one that we wrote here on the left-hand side, but we have added subscripts I for droplet I. They couple to this C infinity field. And of course, we have to add here the Laplace pressure. This is an I, but here the board is a bit. Okay. You see, like this is a coupled equation, which you can simply put on the computer and investigate the growth and shrinkage of many droplets. Now, if that was too fishy for you and you feel like, wait, you did a single droplet, now you just threw it together and made up an equation, that's totally fine. In a way, when you studied Green's functions, you could also do a systematic expansions when droplets are closer to each other and then put them more and more far away and calculate the next leading order perturbation. And then you get simply effects of correlation among droplets. However, this is not relevant. This is just a waste of time to do the mathematics, I would argue, because you just wait a little bit longer in your experiment when droplets are more far away, and they make this equation more and more correct. Yes, this is our model for an emulsion, but it becomes exact if you just wait longer as the emulsion is coarsening. And why is it coarsening? Well, because you have big droplets and you have small droplets. Many of those, as in my left-hand drawing. Now I just draw two for simplicity. Let's combine them here with a line. What happens here at the interfaces? Now, this is now just an illustration, not a real plot, okay? What we have learned is, here this one is a bigger droplet, so this is obviously a larger radius, larger R. But it has a smaller chemical potential here at the interface, so it looks something like this one here. This one here has a smaller radius, which means it has a larger mu bar, or saying it differently, it has a larger Laplace pressure. Okay, which means here the concentration at the boundary is higher. So now you have a lower concentration here and higher concentration and have some sort of gradient here in between. What do the molecules do? Smaller droplet that has at the boundary a higher concentration boundary than the bigger droplet. What happens? So I'm the smaller droplet. Or I'm the bigger droplet. You're the smaller droplet, okay? You have a higher concentration at your boundary. I have a smaller concentration at the boundary. We have a concentration gradient, which increases from my to you. What happens? There's flux, right? Exactly. There's diffusive flux, but from whom to whom? Sorry about that. I could have picked the other example. Exactly, from you to me. Exactly. So which means here down the concentration gradient, so there's flux. which means that the smaller droplet will shrink at the expense of the bigger droplet that grows, while this smaller droplet will eventually shrink. And this phenomena has a famous name. This is called Oswald ripening. So we'll have learned that when you have many droplets, then emulsion is not stable, because an emulsion is composed of small and big droplets. The big droplets, first of all, they have formed, and they have the formation of droplets have pushed down the concentration levels outside close to the equilibrium concentration we obtained from the Maxwell construction. There's no material left anymore. The supersaturation epsilon is tiny. So you cannot grow by just eating up something from your surrounding. You have to kill other droplets. And the only difference between the droplets is their size. Yeah? And how much the difference is exactly characterized or multiplied by the surface tension. So the bigger the surface tension, maybe the faster Oswald ripening will be. We don't know. We haven't calculated it. And then the bigger droplets grow at the expense of smaller ones in a long, long time scales. It's just a single droplet in a finer box of volume B. And this phenomenon is called coarsening or ripening in case of droplets. Yeah? There's an alternative coarsening pathway, and this is fusion. Okay? Because when they're very close by, there's Brownian motion of droplets as well. Then they just fuse and make a bigger drop. Yeah, it's a bit similar. Yeah? Both pathways actually grow the same way in time. It's a matter of prefactor. But fusion we can discuss on a separate note if you like. For the people who find this interesting and think like, okay, what's next? And that's cool for doing numerics. And can I put this in my computer and label my droplets through, etc. And I take them out when they're somehow getting in this regime and about to die. You take them out, dissolve the material, they're dead. There's also a continuum approach here. This is actually what Lifshitz and Sliosov did. I will not do this now. I put this into script. And from this continuum approach, you get this very famous power law that the average radius, because of many radii, you can average of the entire distribution scales with with this famous exponent, and the droplet number scales with 1 over t, in 3 dimensions. Okay, and even more beautifully, the droplet size distribution becomes universal. Okay, this is something I think you have seen universal physics, I guess, in your lectures. The van der Waals gas is one. With different gases, they all collapse to the same universal curve. Here it's even cooler. They all collapse to the same kinetic law, and the distribution in time is universal. You also have to, first of all, a universality in time. I draw it just for the picture, and that you have seen this once. Say you have an early time distribution. It actually you can calculate it. This is the probability of radius r and t. This is the radius. And this is time t1. It looks like this. There are smaller droplets and bigger droplets. Actually, there's a harsh cutoff. You've never seen such a physics distribution, I suppose. You typically see exponentials, Gaussians. This is one on a finite supporter. On a later time, the distribution, if I normalize it, hopefully well enough, it undergoes coarsening. This is t2. This is because it gets to bigger droplets, it increases. And now universality tells you that there is a magic rescaling, actually, with RC, and also this gets to be rescaled, that all distributions at the late time stage collapse to 1. And this calculation you find in my script if you want to have a look. We don't need it for the later lectures necessarily, because this is not what I wanted to present, but also in these active emulsions that are maintained out of equilibrium by mass and energy balance, one can also generalize these concepts from Lifshitz and Sliosov. So if you find this interesting, just let me know. Time to close the lecture for today. We tackled at least kinetics of droplets. We started with a so-called also sometimes phase field approach. We derived the Cahn-Hilliard equation. And this equipped us, after some simplification, it's called narrow interface model, to discuss growth and shrinkage of droplets and even have a simple mind idea of how to implement the dynamics of an emulsion. Yeah? Still, in general, you can always solve what this complicated Cahn-Hilliard equations at time, but as I said, there's not much analytic inside of that. So thanks for listening, and I'm happy to take questions.
So maybe to keep things on time, we can take two questions more or less.
Real quick, we were already making the quasi-state approximation right there at the beginning of the narrow interface model, right? Like the C at equilibrium equal to C0, right?
Exactly.
Okay.
Yeah. So the basic idea of the narrow interface model is, of course, we don't want to remove the interface, and we are left with a boundary condition. That's the first step. Then you could still solve here a nonlinear problem, yeah? But since the gradients are weak, the first step is to expand to up order x, the leading order is linear, and then solve that problem. But with this moving boundary condition, that's different. So compared to a classical electrodynamics problem, it's a moving boundary problem. Yeah? Okay, perfect.
Excuse me, Professor. Could you please explain again that last part of the whole frequency is collapsing? Because I didn't understand very well.
This one?
Yeah.
Yeah, so sorry, this was just an outlook in the end, and the calculation is in my notes. So in principle, so when the system, when you do a probability distribution of an emulsion, it looks at a certain time point, it looks like this. So if you have smaller droplets, you have bigger droplets. There's always the biggest droplet in the system. Yeah? Okay. At a later time, small droplets get kind of, they're less smaller droplets, and you have more bigger droplets. That's this yellow curve. So that's coarsening, yeah? Or ripening. That's fine. That's how it is. And this is, by the way, when you solve this equation, which you obtain in the computer. That's no problem. But now, if you calculate each time point, the critical radius, maybe this is not clear, epsilon is, so C infinity depends on time, epsilon depends on time, thus the critical radius depends on time. So you should picture this distribution moving, this fixed point moving in time. Of course, because it has to eat up all the droplets, it's chasing the system, yeah? Everyone who is too small, I mean, if you're big enough here at that time point, you might not be big enough at a later stage. Only one can survive on long time scales. Yeah? It's a very harsh selection in that sense. But then, when you collapse this, actually with the critical radius, yeah, then there's no time dependency anymore. This is a stationary distribution, and they collapse. Yeah? You can even prove this numerically, just solving that one, yeah, for fun. Yeah?
Okay, thank you.
Okay, so now short coffee break. Thank you again, Christoph.